It's my privilege to give the floor to Professor Aletta Graneveld, who will chair the first session of today. Professor Graneveld.
Thank you, Jan. It's my great pleasure today to introduce this session, which is called Research Update from the International Field, Causes and Diagnostics. And I think we have a nice range of international, very famous speakers. And I'm very happy that I can introduce the first one, which is Professor Anthony Komaroff. He's from the Brigham and Women's Hospital of Harvard Medical School in Boston, US. I think he's one of the front runners in the research in relation to ME-CVS since the late 80s of the last century. And he has sent us a really nice video. Unfortunately, he's not available for questions, but we can maybe ask the others to answer questions in relation to his presentation. So the video can start now.
Hello, thank you so much for inviting me to participate in this important conference. I want to talk today about the research agenda for ME-CFS, both internationally and in the Netherlands. An NIH conference on ME-CFS was held in April of last year. Lasted 2 days, was attended in person and virtually by nearly 5,000 people. There were over 30 research presentations from around the world that summarized knowledge about the underlying biology of the illness and considered the multidisciplinary research agenda going forward. NIH research support for this illness has doubled in the past 2 years, including the establishment of 4 research centers dedicated to the illness in universities, as well as an expanded program at the NIH campus laboratories to study the illness. The summary of what's known about the underlying biology of the illness involved many different ways of looking at the central nervous system and autonomic nervous system. And speakers summarized the neuroendocrine dysfunction, well documented in ME-CFS, cognitive dysfunction, autonomic dysfunction involving both the sympathetic and parasympathetic system, MRI abnormalities, both anatomic and functional MRI abnormalities, SPECT scan abnormalities, PET scan abnormalities, particularly PET scan designed to detect neuroinflammation. We'll come back to that. And finally, EEG abnormalities, both classic type, increased sharp and spike waves, as well as a distinctive spectral coherence pattern of abnormality in the patients. Among the most robust of many immunologic abnormalities that have been found in ME-CFS are increased numbers of CD8 cytotoxic T cells, decreased functioning of natural killer cells, and increased production of pro-inflammatory cytokines and autoantibodies. Perhaps the largest study of cytokines was reported several years ago in the Proceedings of the National Academy of Sciences by Mark Davis from Stanford, one of the world's most distinguished immunologists. They studied nearly 200 cases of ME-CFS and nearly 400 matched healthy control subjects, measuring simultaneously 51 different cytokines in the blood of the subjects. And what they found was that in 17 of those 51 cytokines, the level of the cytokine correlated with the intensity of the symptoms. That for some, a substantial fraction of the cytokines, as the level of the cytokine grew higher, the symptoms grew worse. It was also summarized at the NIH conference that ME/CFS often follows, not always, but often follows an infectious-like illness where doctors haven't actually tried to document what the infecting agent is, but that also it follows very well documented illnesses, infectious illnesses, Epstein-Barr virus infection, Lyme disease, many others. I won't read them all. Perhaps of most topical importance is the growing evidence in the United States and elsewhere that a substantial fraction of patients with COVID-19 appear to be developing a post-infectious fatigue syndrome very similar to, if not identical with, ME/CFS. This unfortunately raises the possibility that we will see a large number, an increased number of cases of ME/CFS in all of our countries in the next year or two. The NIH is very aggressively starting a series of studies to study this phenomenon. In recent years, it has become possible using a variety of techniques of mass spectroscopy to simultaneously measure multiple different molecules in a single sample of fluid, blood. Thousands of molecules simultaneously, for example. What those studies in ME/CFS have shown when the patients are compared with matched healthy control cases is that the levels of most metabolites are lower, as occurs in hibernating animals. That the abnormalities incriminate impairment of cellular energy production and specifically oxidative phosphorylation. That there is possibly a defect in one critical enzyme, pyruvate dehydrogenase, and that there are defects in multiple pathways involved in converting sugars, lipids, amino acids, and of course oxygen into energy. That the organism that feels it doesn't have enough energy has a problem with its cells' ability to produce enough energy. So lots of different underlying biological abnormalities, but what's causing the symptoms? I've only shown one objective abnormality that seems correlated with symptoms, cytokine levels. But an intriguing new hypothesis about what may be causing this illness or the symptoms of this illness is what's called the sickness symptoms hypothesis. And to understand the hypothesis, ask yourself these three questions. What do we feel like when we get the flu? Why do we feel that way? And why do the symptoms go away when the flu has been conquered? The sickness symptoms hypothesis says, is based on the knowledge that humans have a hardwired system that is a temporary acute response to injury or infection, in which the brain orchestrates a set of changes in behavior that are all designed to reduce energy consuming activities. So we become lethargic, we withdraw socially, we become achy, we don't want to move around much, sleepy, we lose our sex drive, we have difficulty thinking, reduced appetite, all changes in behavior designed to preserve our energy stores to fight the infection or the injury that we are dealing with. And that when that infection or injury are healed, the behavior is turned off. This is well documented not only in humans, but in many animals, from invertebrates to even our family pets. I don't think we have much trouble telling when our pets are not feeling well. So what is the hypothesis? It is that these sickness symptoms are orchestrated by a group of neurons in the brain, a fatigue nucleus, that in turn is activated by neuroinflammation. That neuroinflammation may be due to infection or injury of the brain, or it may be inflammation outside the brain that sends signals, both through the blood and through the nerves, as Michael Van Elsecker will talk about, that activates the immune system of the brain when there is inflammation outside the brain. Are there circumstances when this orchestrated response, which kicks in when we get injured or infected and is supposed to be turned off when the injury is done, are there circumstances when that can't be turned off? And there are two, theoretically. One is when we think the inflammation is gone, but there still remains, particularly in the brain, some ongoing neuroinflammation. Another possibility is that the switch, that the neurochemical switch that is supposed to turn off this fatigue nucleus when our stressor is gone, when we've won the battle, that switch gets stuck and doesn't get turned off even though it should. So what's the evidence for the two elements of this theory? That there is neuroinflammation in the brain of ME/CFS patients and that it causes fatigue, and that there is in the brain a fatigue nucleus. And where is it? Well, I have only a very few minutes today, so I can't present all the evidence, only just touch on it. Here is a PET scan from a group in Japan several years ago that was the first evidence of neuroinflammation in people with ME/CFS. What they're plotting here is a marker of neuroinflammation, and in healthy controls, what you see is that the levels are much lower than in patients with ME/CFS. This kind of study has been replicated twice since this first study, but altogether, it's still a fairly small number of patients that have been studied, so we need to know a lot more. But there is some evidence of neuroinflammation in ME/CFS, and there is evidence in other illnesses, particularly a recent study in multiple sclerosis, that the level of neuroinflammation correlates with the intensity of the fatigue, just as the level of cytokines correlated with the intensity of the fatigue. Just a few months ago, in Nature, one of the world's most prestigious scientific journals, two reports were published of a torpor nucleus, the equivalent of what we're calling a fatigue nucleus in mice, that prompts hibernating-like behavior, energy-conserving behavior. In mice, this area was found in the preoptic area of the hypothalamus. No one has yet looked, and it's much harder to look in humans than in mice for such a nucleus, but we're all mammals, and it's not at all implausible that in humans, in the hypothalamus, there may be some similar fatigue nucleus. But please don't misunderstand me in my presenting this attractive hypothesis. I'm not saying that it explains everything about this illness and the symptoms of this illness. I am suggesting that it may be a final common pathway for the symptoms of this illness. There may be multiple different triggers that all lead to a final common pathway of this, that generates the symptoms of ME/CFS. Surely there is also a lot of other relevant pathology in ME/CFS, some of which we've talked about already. The infection or infectious-like illness at the onset, the state of chronic activation of cellular immunity, followed by cell exhaustion among people with longer-term illness, the production of autoantibodies against targets in the nervous system, abnormal energy metabolism with associated oxidative and nitrositive stress, central nervous system involvement as shown by MR, PET, EEG, neuroendocrine studies, autonomic dysfunction that causes reduced blood flow to the brain, neuroinflammation, dysbiosis involving the microbiome of the gut that possibly is a chronic stimulus to neuroinflammation, and calcium signaling abnormalities. So what is our research agenda, in my opinion, for the Netherlands and for the world? It's to understand all of those abnormalities I just mentioned a little better, but above all, to find a pathogenetic hypothesis that explains some or all of these different observations. How are all these abnormalities related to one another? Which one comes first? Is it in fact possible that several of them could come first and cause a set of dominoes to fall, other systems to become disordered, regardless of which system became abnormal first? These are intriguing possibilities to pursue, and in my opinion, represent the best, most focused kind of agenda for ME/CFS research moving forward. So thanks again for having me participate in the conference, and I hope I've stimulated some thoughts.